Okay. Good afternoon. I'm going to take that chapter name is Generation and Conduction of Impulses. Generation and Conduction of Impulses. What do you mean by generation and conduction of impulses? We already studied that the structure of neuron. So in structure of neuron, there is what a synaptic knob is that we know the dendrites. That is, first of all, the stimulus is collected by dendrites and it is inside the cytone. It makes us what action potential. Then it is travels through axon, finally ending to axonites or synaptic knob. This is the way of transmission of impulse. So we are receiving a stimulus, and that stimulus is converted as an impulse, then only transmitted through this axon. So how these impulses is transmitted? Through axon, so that we can study. First of all, neurons are the excitable cells. What do you mean by excitable cells? Excitable cells means during resting time also it is in polarized state. For example, so this is the axon. So inside the axon we can call it as axoplasm, and this membrane is axolemma, and outside is called as extracellular fluid. Okay. So this neuron is in resting state. It is a charged state. How it become charged? Inside the axon there are ions. Ions are there. Outside also ions are there. So here a gradient is there. What is gradient? It means one place it is higher concentration and other place it is lower concentration. That is called as gradient. So inside the axoplasm there are two types of ions are there, that is sodium and potassium. So outside also, that means in extracellular fluid also sodium and potassium is there. So this potassium ion is more in axoplasm and less in extracellular fluid. That means potassium ion is present more in axoplasm. That is, when compared to this, it is less in outside membrane. That means in extracellular fluid. So that is a gradient. Like that, the sodium is more in extracellular fluid and it is less concentration in what? Axoplasm. So that is a gradient. So what is there? Na plus is more. That means sodium is more in extracellular fluid and potassium is less. So like that, inside the axoplasm, sodium is less and potassium is more. Okay, so there is a gradient. And also inside, that means in axoplasm, negatively charged proteins also present. Okay, understand? There is the what? Gradient is there. Then other thing, this gradient is what? The ions are in higher in what? Sodium is higher or where in the outside and low in less concentration in axoplasm. Like that, potassium is what? Less in extracellular fluid and more in axoplasm. So there is a gradient. And also there are different type of ion channels are there. Different type of ion channels to move. That we have already studied that wherever the ions are in higher concentration, that move to lower concentration for what? Lower concentration through the semi-permeable membrane, that we already know. That is a movement of ions. So for this movement, they are having certain channels are there. There are three types of channels are present. That is, one is what? Leaky channel. Leaky channel means what? There is no need of any voltage or anything. There is no gatekeeper is there. The ion can easily move from higher concentration region to lower concentration region. The other channel is called voltage gated channel. Voltage gated channel means what? The channels which there is a voltage, they need certain voltage or there what? If any stimulus is there, then only this channel should be opened. So that is called voltage gated channel. The other one is pump. We already studied what is the function of pump. Pump allows the particle from lower concentration to higher concentration. That means what type of transport takes place there? That is active transport. Pump is actively transporting, and leaky channel and voltage gated channel are passive transport. What do you mean by passive and active? Passive means there is no need of energy. Particles move from higher to lower. That is called passive transport. Active means the particles or ions move from lower to higher with energy expenditure. So pump is actively transporting ions, and leaky and voltage gated channel are passively transporting ions. So you know what are the three type of channels and what is the ion gradient inside the exoplasm and also outside the exoplasm, that means in extracellular fluid. So when then now we can study what happens during resting period. During resting period, that means in resting time. So in extracellular fluid, that is, this is the exoplasm. That means there is no stimulus is transmitting. During that time, exoplasm and in extracellular fluid, we already know in exoplasm there is more potassium, less in sodium and negatively charged protein. In extracellular fluid, more sodium, less potassium. Okay. So during the resting state, this leaky channel, that means potassium leaky channel is open. Leaky channel means there is no need of gatekeeper. We know we don't want any permission to go inside or outside. So like that. So there is leaky channel open during resting period. That means potassium leaky channels. So the exoplasm contain more amount of potassium. No? This potassium move to extracellular fluid through this leaky channel. So that means potassium is moving outside during resting stage. And negatively charged proteins are impermeable, that is inside only. And what happens if the potassium is moved from exoplasm to extracellular fluid also? That is means what? It is limited amount. But in the case, what? There is another pump is open here. Pump is open. That's a pump. What is the pump? What type of movement allows? That is passive that means active transport. That means that is the higher particles move from low to high. Is it? So here sodium is less, potassium is more. So sodium moves outside, and here potassium is less and sodium is high. So potassium moves inside. Okay, so there is what happens. Already the potassium ions are moving exoplasm to extracellular fluid, and in this pump also allows the movement of positively charged ions to the outside. So what happens here? Potassium comes inside and sodium moves outside. So there are 3 sodium moves outside, then 2 potassium comes to inside. So what happens? Mainly already potassium ions are going to outside, and there's some in this way the pump opened, and again when the 3 sodium goes outside, only 2 potassium comes inside. So like this way what we understood, that means more positive ions are going outside and less positive ions are comes inside. So already we know wherever the positive ions are there, along with that negative ions also there. So here more positive ions are going outside. So what happens here? Inside, that is more negative ions are remaining. That means more positive ions are going. So the some negative ions are what? Along with this positive ions. So we lost the positive ions that goes outside, and the negative ions are inside. Already negatively charged protein is there. Along with that negative ions also alone. What happened? We know already studied what? Chloride is shift. Is it? That is chloride is there. This chloride is shifting here and there. That is negative ions. So here also along with this potassium or sodium, there are some negative ions also there. When more positive ions are going outside, that means their companion negative ions are along here. That negative ions are making a charge inside the exoplasm, that is negative charge. Outside more positive ions are there, so outside it is positive charge. This charged difference is called as what? It is a what? Resting potential. Resting potential. That means during resting state, that is ions are moving or what? Through exoplasm to outside. So there is a negative charge inside the exoplasm and positive charge is outside the exoplasm. That is called as what? That is the resting potential. Okay, that means more potassium ions are going outside, is it? And also along with that, here also more sodium is going outside. So more positive ions are goes outside and less, that is what, positive ions are coming through this pump. So that there is a negative ions are there, that makes a charge inside the exoplasm is negative charge, and more positive ions are outside, that makes a charge called as positive charge. So there is extracellular fluid is in positive charge and exoplasm is in negative charge. This charged difference is called as what? That is called as resting potential. Understand what is resting potential? There is during resting time there is a charge difference. So what we have to conclude finally, in the exoplasm there is mainly high concentration of potassium and also less concentration of sodium and negatively charged protein. In extracellular fluid, high concentration of sodium and less concentration of potassium. So what happens then when in the resting time, more positive ions are goes outside through the leaky channel as well as in pump. Pump is mainly acting this polarity. Okay, so there is what? More positive ions are goes outside and less positive ions are comes inside through the pump. So that way there is morely negative ions are inside the exoplasm. So there is a negative charge and outside it is a positive charge. That charged difference is called as resting potential. That is called as resting potential. Understand? During resting time, there is a voltage is minus 70 millivolt. It is called as resting potential. Understand? And the next one is how the stimulus is passed. When stimulus passed from the what? Passed through this axon, what happened? That means action potential. So we know already that is what during the resting time it is outside positive charge and inside negative charge. When the voltage is applied here, so voltage means a stimulus or an impulse. Impulse is passed during that stimulus is called threshold stimulus. That means they needed a minimum voltage. Okay, minimum voltage is needed, and during this when they have this threshold stimulus or when they get a stimulus, what happens? Suddenly the voltage gated channel, sodium voltage gated channel is opened. When the sodium voltage gate channel is open, outside we know already what is there outside the hole, that is positive ions, like sodium is there more in outside. So this sodium are what? When the stimulus is, this is a fraction of second, this voltage gated channel is opened only in a fraction of second. So during that time when it opened, outside there is a positive ions like sodium, and that sodium is high influx of sodium to the exoplasm. Suddenly we know that is the door is going to close within a fraction of second. So what we do, we simply push up and we will go inside. So like that the sodium ions are more in outside, this voltage gated channel is open and that will close just in a second, within a second. So for that time, what happened? This sodium is pushed and come inside. The high influx of sodium into the exoplasm, that is called as what? That is more sodium is comes inside. That means what? More negative ions are alone in the outside. So what happens? Inside there is in our exoplasm, there is a positive charge and outside it is negative charge. Understand? So this is the action potential. So during resting time, outside positive and inside negative, that because of the pump. Okay, and during the action time, that means when the stimulus is applied, what happens? Outside negative and inside positive. How it happen? Because of the sodium voltage gated channel is open and suddenly the sodium ions are in highly what enter high influx of sodium into the exoplasm, so that more sodium ions comes to the exoplasm. So exoplasm contains positive charge and outside it is negative charge. That is called as what? That is That is, sodium also move from through water, that is during passive transport only. So like this way, what happens mainly, the charges changed. Okay, that is called as what? Action potential. So the stimulus means what we are getting, that is stimuli. Light is a stimuli, and even the what, even the sound wave is a stimuli, and also that are the external stimuli, or chemicals, temperature, everything is a stimuli. When we get a stimuli or a stimulus is what, in a what particular temperature, that is called threshold stimulus. Okay, threshold stimulus is minus 60 millivolt. So during that time, and when it what, during as stimulus applied here, what happened? Voltage gated channel open and sodium comes inside and outside. That means in exoplasm positive and outside negative charge. That is called as depolarized state or action potential. So in the resting time, what happens? Outside positive and inside negative. That means outside the axon it is positive charge and inside the axon it's negative charge. That is called resting potential. During action time, when they get the stimulus, outside negative and inside positive charge. That because of sodium what, voltage gated channel. That means sodium comes inside. That's why this charge is different. Understand? So that is called as action potential. So now we can know what is transmission of impulse. So impulse is transmitted and it is reaches to one side. So how it is transmitted from one region to other, that we can study transmission of impulse. Now we understand the charge difference and all. So now we can study how this different, that means this will be transmitted. So this is the side A and this is B and this is C. So here it is during resting state, outside positive, that means inside negative. Outside positive, inside negative, outside positive, inside negative. When a stimulus passed here, what happens? The charge difference, charges, what happens? Outside negative and inside positive. How it happen? Because of sodium influx. Understand? So this stimulus suddenly transmit from this side to this side. Okay, when the stimulus reached here, this charge difference is changed. Here it is outside positive and inside, sorry, outside negative and inside positive. So how it happened? That means the sodium, potassium. So like that time, what happened? Here there is a resting state. When the stimulus passed to side B, the side A is in a repolarized state. What is repolarized state? That means it is in a resting state. That means what? Outside it is positive and inside it is negative. That we know how. That means the positive ions are comes here and here the positive. That means here what happens? Now you understand, in the side A there is a action potential. That means outside negative, inside positive. When it comes to the side B, already here how it is the side is outside positive and inside negative. So what happens? This positive ion moves here and this positive ion is moves here. That is we know. Positive ion is moves towards the negative. So this is the completion of circuit, is it? So how it happens? That means how it happened? This positive comes here, also that this time here it is negative and this positive goes here and here it is positive and here it is what? Negative. So how it happens? That is because of completion of circuit, that is repolarized state. That means when a stimulus passed from side A to side B, the side A become in a resting state. Understand? Resting state means what? The sodium pump is open and sodium comes inside, then exoplasm is positive and outside it is negative. Understand? So when the stimulus is passed to side B, what happened? In the side A, the sodium pump is closed and potassium leaky channels open and potassium ions move into exoplasm and also pump also open. Again sodium comes, sodium goes from exoplasm to outside and potassium from extracellular fluid to exoplasm. That what resting state, that is called as repolarized state. So here it become repolarized. So when the stimulus passed here and here it is outside negative and inside positive. How it is? Because of here side B is repolarized and side C is depolarized. So like that, stimulus passed from one side to other side. That means whenever the stimulus passed from side A to side B, side A become repolarized and side B become depolarized. Again side B to side C, side B is repolarized and side C is depolarized. Why it is happens? Because our body, the stimulus is passed one by one, very fast mode. You see, when I am taking class, I am receiving, or I am my sensory organs are receiving the stimulus and suddenly responding. So stimulus is passed from one region to other in a very fast mode. So that when the stimulus passed from side A to side B, the side A become what? Repolarized and side B become depolarized. When it is goes to side C, then side B become repolarized and side C become depolarized. So on that time, side A is start to depolarize because another stimulus is comes in back of this. So like that, one by one, suddenly the stimulus is passed so that the older region, that means the stimulus, that is the side A region, become repolarized. Understand? So this is the way this stimulus passed through this axonal membrane and then it is reached to the synaptic knob. Synaptic knob means that is when there is an axonites. We know axonites. A bulb-like region is called as synaptic knob. This synaptic knob, this what stimulus is passed. There are two types of impulses are there. One is chemical impulse, the other one is electrical impulse. Chemical impulse means when a chemical which is reached to the synaptic knob. So in our body there is a chemical transmission. But in electrical impulse only in the electric ray that fishes and all, that electrical impulse is there. That is like what? When we on the switch, the light is on. Is it? So that is very fast mode. The impulse is transmitted from one, that is means axon and synaptic node to other dendrites. Is it? Presynaptic neuron means that is a neuron which present what? Before the synapse. Postsynaptic means after the synapse. So here the what? That is electrical impulse passed from one neuron to other in a fast mode directly. That means there is a junction is there, cell junction or gap junction. We know gap junction means what? There is a what? That means there is a connection between two cells that is called as gap junction. That means for the communication of substances we already studied. So here when the electrical impulse passed from presynaptic to there is no synapses there. Synapse means that is what? That is there is no space between two what? Two adjacent neurons. So that impulse is passed directly from one neuron to other. There is no cell junction or that is there is no what? Synapse is there in between. There is no junction is there and it is directly passed and that is also it is bidirectional way. That is takes place only in lower cat organisms. So we will we will get a doubt that all the advanced changes or development are for human being, then how we are in chemical transmission and that is lower fishes are in electrical transmission. That means during electrical transmission, one impulse transmission, in between two transmission, there is a what? Preparation is needed for a neuron. That means the stimulus is not what? Directly, whatever, if one stimulus is passed and then if the next, when they receive another stimulus, there is a gap. They need a gap for preparation. So in our cases, we can't take a break. Is it? Then when you ask a question, I will take a few minutes to think, and after that, I will talk one word again after a preparation. Then only that means when stimulus pass, then only I can talk. Is it? But in the case of lower organism, no need of that much advanced transmission is takes place, so that there is an electrical transmission. That means there is no cell junction. Directly the impulse is passed from one neuron to other neuron, like a switch. What we on the switch, the light is on, like that way. But in the case of chemical transmission, the synaptic knob, inside the synaptic knob there are certain chemicals. We already studied that in neuron there is a what? There is a cell, a particle called as what? In a neuron there is a particle which is called as Nissel's granule. The Nissel's granule only producing a protein called as neurotransmitter. That neurotransmitter like acetylcholine that reached to the synaptic knob through the impulse transmission. When this this is when this acetylcholine, that means when the impulse is reached to the synaptic knob, what happens? Outside there are calcium ions. These calcium ions comes inside the synaptic knob, and what the synaptic vesicles are fused with the synaptic vesicle membrane. It is fused with the membrane of the synaptic knob. Knob membrane is fused with the vesicle membrane. During that time, the calcium ions are binds this vesicles, and when this binding takes place, this membrane, both the membrane, that means vesicle membrane as well as the knob membrane, both membranes are fused and burst. For this fusion, it bursts and release the chemical transmitter in the synaptic junction and when there are synapse, and then these chemicals are binds with the dendrites of postsynaptic neuron. Against this, the impulse transmission is carried on. So this is the way impulse transmission takes place. Understand how the chemical transmission takes place? When the impulse reached to the synaptic knob, there is a calcium ions enter inside the synaptic vesicle, that is synaptic knob, and it is binds with the synaptic vesicle. During this binding, the vesicle membrane and knob membrane fuse together and burst it, release the chemicals in the synapse. Then that chemicals are binds with the dendrites of postsynaptic neuron. Understand? So this is the way the impulse transmission takes place. Okay, this is the way of transmission of impulses. So we know how first, that is a resting time, as well as first we study what are all the points we should remember from here. It is what is a very important chapter for NEET, and also what are all the things happens, you can summarize there. So first thing, what happens? First thing, during transmission or impulse transmission, what happens? Yes. Yes, first of all, during resting time, that is the charge is in axoplasm, we know outside positive and inside negative. How it takes place, how it happens? Because of the leaky channels open, so potassium ions goes outside, and also pump open, and less potassium comes inside and more sodium goes outside. Because of that, more positive ions are lost from the axoplasm to the extracellular fluid, so that here it is outside positive and inside negative. That is called as that is the resting potential. That charge difference is called as resting potential. Next, action potential. What happens? During the action time, there is a sodium pump. So sodium voltage gated channels open. More sodium ions are comes inside. That means outside there is more sodium ions. That all sodium ions are comes inside. So outside was negative and inside positive. That is called as action potential. Okay, that is action potential and resting potential. Then how the impulse transmission takes place? When the impulse is applied to the site A become outside negative and inside positive. When it goes to the site B, and site A become outside positive and inside negative, and site B become outside negative and inside positive. So like that way, the impulse goes out into the site C, site D, like that. It reached to the axonites. When it reaches to the axonite, there is a synaptic knob. Inside the vesicles are there. The vesicles are filled with the neurotransmitter. And when the charges reached to the synaptic knob, calcium ions comes inside the synaptic knob, and it is binds with the vesicles, and the vesicles are moves towards the knob. Knob membrane, and knob membrane and vesicle membrane are joints are fused and release the chemical transmitter called as acetylcholine, and it is binds with the dendrites of postsynaptic neuron. Understand? This is the what? Transmission of impulses. Understand all of you? And this is transmission of impulses. That is very important topic in neural control and coordination. So you will get definitely you get questions from this chapter. Okay, definitely you get 1 or 2 questions for NEET. It is a definite question also. Okay, thank you all and what? Thank you.